Hello everyone, welcome to our channel Biological Concepts. In today's video, we will going to discuss on the differences between gram-positive bacteria and gram-negative bacteria. So stay tuned. Before going to the details on the differences between gram-positive bacteria and gram-negative bacteria, let's know from where this gram word is taken. There was a Danish bacteriologist named Hans Christian Gram, and from his name the gram word is taken. He developed the gram staining technique in 1884. The bacteria which are able to retain this gram stain known as the gram-positive bacteria, and which are unable to retain this gram stain known as the gram-negative bacteria. Now let's discuss the differences on this gram-positive bacteria and gram-negative bacteria in details. First is the cell wall. The cell wall composition of gram-positive bacteria and gram-negative bacteria is different. In case of gram-positive bacteria, it consists of 20 to 80 nanometer thick homogeneous layer of peptidoglycan. This peptidoglycan, also known as the murin. This peptidoglycan layer is lying outside the plasma membrane. So, in case of gram-positive bacteria, the homogeneous layer, that is peptidoglycan, is 20 to 80 nanometer thick. Next is the gram-negative bacteria. The cell wall composition of the gram-negative bacteria is quite complex. It is covered by an outer membrane. It is first consists of the peptidoglycan layer, which is covered by a thick outer membrane, which was not present in the gram-positive bacteria. So this peptidoglycan layer is 2 to 7 nanometer thick, whereas in gram-positive bacteria it is 20 to 80 nanometer thick. And this peptidoglycan layer is covered by a thick outer membrane which is 7 to 8 nanometer thick. Next is the presence of outer membrane. The gram-positive bacteria, the absent is outer membrane. In gram-negative bacteria, the outer membrane is present. Next is the presence of peptidoglycan layer. In case of both gram-positive bacteria and gram-negative bacteria, the peptidoglycan layer is present. In case of gram-positive bacteria, it is thick and multi-layered, whereas in case of gram-negative bacteria, it is thin and single-layered. Now the presence of teichoic acid. The teichoic acid is present in gram-positive bacteria. In gram-negative bacteria, it is absent. The teichoic acid is basically a acidic polymer of ribitol phosphate or crysalyl phosphate. It gives the bacteria negative charge. When this teichoic acid is present attached with the plasma membrane, it is known as the lipoteichoic acid. Next is the lipid content. In case of gram-positive bacteria, the lipid content is very low. In case of gram-negative bacteria, they contain high amount of lipid content. Next is the presence of lipopolysaccharide, which is shortly known as the LPS. This LPS layer is absent in gram-positive bacteria, but in gram-negative bacteria, it is present. Possibly the most unusual constituent of the outer membrane are its LPS, that is the lipopolysaccharides. They are quite complex molecules and contain both lipid and carbohydrate and consist of three parts. One is lipid A, second one is core polysaccharide. Polysaccharide. This is one, this is two, and number 3 is the O side chain. O side chain. Okay. Now, as the outer membrane is absent in gram-positive bacteria, so it is also the LPS is also absent in case of gram-positive bacteria, because this LPS is the unusual constituent of this outer membrane. Now, next is the periplasmic space. The periplasmic space in gram-positive bacteria is smaller than the gram-negative bacteria. In gram-negative bacteria, this periplasmic space is larger. than the gram-positive bacteria. Next is the color retention. Color retention on gum staining. The gram-positive bacteria retain the crystal violet, which is the primary stain, and appear purple under this microscope. The gram-negative bacteria do not retain the crystal violet, that is the primary stain, and appear pink under the microscope. Here the steps of the gram staining procedure is briefly mentioned. First, the primary stain crystal violet is added. After addition of this crystal violet, that is the primary stain, the cells of the bacteria stain the purple. Next, the modern iodine is added, which form the CVI complex, crystal violet iodine complex, and the cells remains purple. After the third step is the decolorization procedure, that is done by the addition of the alcohol. During this decolorization procedure, the in gram-negative bacteria become colorless and the gram-positive cell remain purple, because the alcohol dissolves the lipid content of the outer membrane of gram-negative bacteria, and their outer membrane is dissolute. As a result, they dissolves this primary stain color, that is crystal violet, and become colorless. But in case of gram-positive bacteria, on addition of the alcohol, the peptidoglycan linkage is become more cross-linkage is greater upon addition of the alcohol, and the CVI complex more tightly bound to this peptidoglycan layer, and as a result they retain this crystal violet color, which is the purple color. Number 4 is the safranin, that is the counter stain addition. On addition of the counter stain, gram-positive cells remain purple because the CVI complex does not remove from their cell. But in case of gram-negative cells, they appear red, that is take the counter stain colored safranin. Because on addition of this alcohol, that is the decolorizer, their outer membrane is dissolved from their membrane, and they remove this crystal violet iodine complex, that is the purple color, and become colorless. On addition of this safranin, that is the counter stain, they appear red or purple pink in color. Next is the characteristics on resistance or susceptibility on different kinds of thing. The first is the resistance to osmotic pressure. The gram-positive cells are more resistant and the gram are less resistant. Susceptibility to antibiotic. They are highly susceptible because they does not contain the outer membrane. But the gram-negative cells are less susceptible to antibiotic because of their presence of outer membrane, which act as a barrier. Number 11 is the susceptibility to anionic detergent. Gram-positive cells are more susceptible to anionic detergent, but the gram-negative cells are less susceptible to anionic detergent. This is also because of the presence of outer membrane, which act as a barrier. Okay. Type of toxin produced. The gram-positive cells produce exotoxin, whereas the gram-negative cells produce endotoxin. This production of endotoxin is also due to their presence of the outer membrane. Now the examples. The examples of gram-positive bacteria are Staphylococci, Streptococci, Lactococci, Bacillus, Clostridium, etc. And the examples of gram-negative bacteria are E. coli, Shigella, Salmonella, Vibrio, Helicobacter, Pseudomonas, etc. One video on the easy way to remember the gram-positive and gram-negative bacteria has already been uploaded in this channel. The link will be shared in the description box. Thank you. These are all about the differences between gram-positive bacteria and gram-negative bacteria. If you like this video, please like, share and subscribe to this channel. Thank you.